My name is Bango and today in this video lecture we will see the topic molecular basis of inheritance. So first of all we will start with the experiments that led to the identification of the genetic material. So the first experiment we will be seeing is the Griffith experiment. First we will see the Griffith experiment. So Griffith conducted his experiment in the year 1928. He used the two organisms. First was the E. coli and second was the mice. So the bacteria that was used is the Streptococcus pneumonia. Now this Streptococcus pneumonia, when it infects the organism, it has the ability to cause the pneumonia in that organisms, and eventually the death of this organism occurs. So this Streptococcus pneumonia has two types of colony. Colony is the group of bacteria that have the similar properties and they live together. So this Streptococcus pneumonia has two types of colony. First colony is the smooth colony, and second type of colony is the rough colony. Now the smooth colony have the ability of the virulence. Virulence is the ability of an organisms or the bacteria to cause the disease. So this smooth colony was virulent. It was able to cause the pneumonia in which it is going to infect. And this rough colony do not have the ability to cause pneumonia, so it is called as avirulent. So the strain of this smooth colony that was used is the 3S type. 3S. And for the rough, the strain used was the 2R. So first was the Streptococcus pneumoniae and second he used the mice. So the Streptococcus pneumoniae was injected in the mice, and now we will see how the experiment was conducted by the Griffith in year 1928. So in the first step, he took the bacteria 3S, which was in living condition, and this is virulent, has the ability to cause the pneumonia in the organisms. So here the organism used was the mice. So this is mice, and due to which after the infection, all the mice that were infected, all mice died due to the pneumonia. So now in the second step, he took the same 3S bacteria, but that was heat killed. Now all these bacteria are dead, so they have lost their ability of the virulence. So they will not produce any type of pneumonia in the mice. So when they were infected in the mice, no mice died. So the third experiment he conducted, the third type is that he took the 2R bacteria. Now this 2R bacteria, it is a avirulent type and it has no ability to cause any type of pneumonia. So it is avirulent. Now when it will be infected or injected in the mice, all mice will live, as it will not cause any type of pneumonia. So these were the basic three steps. Now the fourth step is the interesting step. So in the fourth step, he took the 3S bacteria that was heat killed. Plus, he took the 2R bacteria that was in the living condition. Now these 2R bacteria do not cause any disease, and this heat kill bacteria also has lost its capacity to cause disease. So when these both were infected in the mice, some mice died. Now, the mice that were dead, he took those mice and he extracted the cells from these mice, and these cells were observed, and in these cells live 3S live bacteria was found. So what Griffith concluded is that there is one component in 3S, a component from 3S, it transformed the bacteria 2R, which made this 2R virulent. And he named this component as the transforming principle. Transforming principle. And he concluded his experiment. Now, in year 1944, Avery, McLeod and M. Carty conducted the experiment to access the biochemical nature of this transforming principle. So we will see the experiment conducted by Avery and co-workers in year 1944. So the experiment conducted by Avery and co-workers in 1944 were in vitro. This experiment tells us about the biochemical nature of transforming principle. While doing their experiment, they discovered the protein digesting enzymes called as the protease. They discovered the protease. Also, they discovered the RNAs as well as the DNAs. Now, what are these protease, RNAs and DNAs? So, protease are the enzymes that have the capacity to digest the protein. Now, these RNAs, they will digest the RNA. And the DNAs will digest the DNA. And they will not have any type of other actions on any other. This protein will not act on RNA or DNA. Similarly, RNAs will not act on the protein or the DNA, and DNA will not act on the proteins and the RNA. They have only specific work, that is DNAs will only digest the DNA, RNAs will only digest RNA, and protease will only digest the protease. So, after conducting their experiment, Avery and co-workers concluded that the biochemical nature of the transforming principle of Griffith is the DNA. So, after Avery and co-workers, in 1952, the experiment were conducted by Hershey and Chase. Experiment of Hershey and Chase in year 1952. The experiment conducted by Hershey and Chase led to the universal acceptance of DNA and genetic material. Now we will see how the experiments were conducted by the Hershey and Chase. So Hershey and Chase used two organisms. First is the E. coli. This E. coli is the bacteria. It is a bacteria. And the second organism used is the T2 bacteriophage. Now this T2 bacteriophage, don't get confused in the name. This bacteriophage, it is a virus. This virus infects the bacteria. Virus that infects the bacteria. Bacteriophage is the virus that infects the bacteria. So this bacteriophage has a very simple structure. We will now see the structure of this bacteriophage. Bacteriophage is composed of only two components. First is the DNA and second is the protein code. So this is the structure of the bacteriophage. It has three pairs of legs, in total 6 legs. This is the outer protein code. And in this protein code, the DNA is enclosed. This in the blue color, it is the DNA of the virus. Now when this T2 bacteriophage infects the E. coli, it inserts the DNA in the E. coli. It will insert the DNA in E. coli when it will infect the E. coli. Now we will see how the experiments were conducted by Hershey and Chase. Hershey and Chase also used the radioactive sulfur and radioactive phosphorus. We know that the protein is composed of sulfur and the phosphorus is a component of the DNA. So when these radioactive compounds are emitted to UV radiation, they emit the light. These radioactive compounds emit the light, or they glow in UV rays or UV light. Now we will see how they conducted the experiment. Now some of the bacteriophage were given nutrient media that is rich in the sulfur, radioactive sulfur, and normal phosphorus is also present in there, but the majority of portion is of radioactive sulfur. And some of the bacteriophage were replicated using the radioactive phosphorus. Here red color shows the radioactive phosphorus, and normal sulfur was also present. So now this T2 phase were allowed to replicate. Some T2 phase were allowed on this and some were allowed on the radioactive. This is the phosphorus and this will be our radioactive sulfur. Now what is going to happen is, when the T2 phase that were grown on the radioactive sulfur are going to uptake the sulfur, and only in their protein code we are going to find the sulfur, radioactive sulfur, and the bacteriophage that were replicated in the radioactive phosphorus media, their DNA part is going to uptake this radioactive sulfur and not the protein code. So now this protein, after their multiplication, this T2 phase were allowed to infect the bacterial cell. Now after their infection, this is the radioactive DNA, and here it will be the radioactive sulfur, that is the protein code. Now both the infection will occur. Now this DNA will be inserted in this E. coli. After its insertion, the only protein code will remain outside this cell, and the same here, this DNA will also be injected in the bacterial cells. Now this E. coli will multiply the viral DNA in both the cases. After their multiplication, now this T2 phase bacteriophage is attached to the E. coli cells. Here also it is attached. Now her scientist took this complete culture in a centrifugation machine. So after centrifugation, what happened? That the lighter portion, which will be the protein coat, and the heavier portion, that will be the E. coli cell. The E. coli cell settled at the bottom. These are our test tube, and these are the normal E. coli cells. They will now settle at the bottom, and the supernatant, which is the lighter part, will float on the upper side. Similarly, for this also, the bacterial cells will accumulate at the bottom as they are the heavy portion, and the supernatant will be floating. Now when this complete mixture was exposed to the UV radiation, there was radioactivity found in the supernatant. Radioactivity in supernatant, which confirms that the proteins were present outside the cells. And when these were exposed, the radioactivity was seen. Radioactivity was seen in the cells, which confirms that the DNA was the portion that was inherited, or that was used to infect these E. coli cells. And the experiments of Hershey and Chase led to the universal acceptance of the DNA as the genetic material in majority of organisms. Now we will see some organisms that have the genetic material of some organisms. So animals, all animals like humans, tigers, cat, dogs, plants, bacteria, most of the animal virus, and most of the bacterial viruses. All these will have the DNA as their genetic material. DNA as genetic material. Now, and in this here we have to add cauliflower mosaic virus also. Cauliflower mosaic virus. This cauliflower mosaic virus is a plant virus, which you have to mention specifically because questions can be asked from this, that cauliflower mosaic virus has the double-stranded DNA as their genetic material. So these are the organisms that have the DNA as the genetic material. Now there are some organisms which are having RNA as their genetic material. Now we will see the organisms that have the RNA as their genetic material. Now the first organism, that is the tobacco mosaic virus. Tobacco mosaic virus. After that, plant wound and tumor virus. Now the viruses such as influenza, rogue sarcoma, encephalitis, and reovirus. These are the organisms that have RNA as genetic material. So these are the organisms that we have to consider: tobacco mosaic virus, plant wound and tumor virus, influenza virus, Rouse mosaic virus, sarcoma, encephalitis, and reovirus. They are having the RNA as their genetic material. Now, after seeing the organisms and their genetic material, now we will see the properties of the genetic material, or the features of the genetic material. Features of genetic material. So, very first is the genetic material should be able to replicate itself. Now, replication means to multiply. That is, it should be able to make its copies so that in the next generation it can give the copies to its offsprings. Second property is that it should be structurally and chemically stable. Chemically it should be stable. The third property is that there should be small amount of genetic material should allow small amount of mutations in them. So what are these mutations? They are the sudden heritable changes. These sudden heritable changes are required for the variations, and these variations will help the organisms to survive in the environment and or in the adverse conditions. And the fourth is it should follow the Mendelian laws of Mendelian inheritance. So these are the properties or the features of the genetic material. That is, first, it should be able to replicate itself. Second, it should be stable and chemically stable. Third is that it should allow the small amount of variations. And fourth, it should follow the laws of the Mendelian inheritance. Now, after the features, we will move forward to the components of the DNA. Now, the components of the DNA. DNA is the polymer. It is the polymer that is made up of nucleotides. So these nucleotides, they are the basic units of the DNA. So this nucleotide is made up of three components. First is the sugar, second is the nitrogen bases, and third is the phosphate group. Now, these are the components of the nucleotide. First is the sugar, second is nitrogen base, and third is the phosphate group. Now, when the bond is formed between the sugar and the nitrogen base, sugar plus nitrogen base, this is called as the nucleoside. It is the nucleoside. Now, when this nucleoside plus phosphate group, they form the bond between them. This leads to the formation of the nucleotide. So, sugar plus nitrogen bases is going to give the nucleoside, and when this is the nucleoside, there is addition of the phosphate group, it is going to give us the nucleotide. Now, we will see the structure of this all sugar, nitrogen bases, and phosphate group. The sugar present in the DNA is the deoxyribose sugar. This sugar is a pentose sugar. That is, it is made up of the 5 carbons. 5 carbons, and there is a one oxygen molecule. Oxygen. And this is the 5 carbons. Now these 5 carbons, out of these 5 carbons, 4 carbons are in the cycle. They are the cyclic. And this one carbon that is left, that is present outside the cycle. Outside cycle. Now we will see the structure of this pentose sugar. So this is our oxygen molecule. Now this is the pentose, composed of 5 carbons. This will be our first carbon, second carbon, third carbon, fourth carbon, and the 5th carbon, which is present outside the cycle. And this will be our 5th carbon here. This will be our 5th carbon. Now there are two bonds will be free, as carbon has valency 4 valency. The 2 valencies are completed by this bond and the bond with the other. So the 2 valencies are free. So there will be presence of H and OH. And the second carbon, there will be only hydrogen and hydrogen. While at the third, there will be H and OH both. And at the there will be CH2OH. Now this is the structure of the sugar that is present in the DNA. This is the 2-deoxyribose sugar. In the ribose sugar, there is presence of the oxygen molecule here. The oxygen molecule is present in the ribose sugar, but it is the sugar that is present in the DNA, so it is the deoxyribose. The removal of this oxygen molecule leads to the formation of the deoxy sugar. So after this removal, this is the 2-deoxyribose sugar. Now after the structure of sugar, we will see the structure of the phosphate group. Phosphate group. This phosphate group, the formula for phosphate group is H3PO4. There is a phosphate present. It has. Now there are 4 oxygens. One oxygen will be present in the double bond with the phosphorus, and other 3 valencies are present now out of the 5 valencies. Now each this will be satisfied by the oxygen. This valence will be satisfied by the oxygen, and now this oxygen, there will be the presence of the hydrogen. 3 hydrogens will be present. So this is the simple structure of the phosphate group. Now after the phosphate group, we will see the nitrogen bases. Third is the nitrogen bases. The nitrogen bases that are present in the DNA are of 2 types. First is the pyrimidines, and second are the purines. Now these pyrimidines, they are the single ring. Of 6 membered. And these purines are made up of double ring and are composed of 9 members. So in these cyclic rings, there is nitrogen present. So the nitrogen in this pyrimidines is present at the 1 and the 3rd position. So at the 1st and 3rd position, nitrogen is present. Only 2 nitrogens are present in this pyrimidine. While in the purines, nitrogens are present at 1, 3, 7 and 9th position are the nitrogens. So out of 9, there will be 4 nitrogens. Now we will see the types of pyrimidines and purines. The pyrimidines are of 3 types. So the first is the uracil, second is the thymine, and third is the cytosine. Now this uracil, thymine and cytosine are the pyrimidines, and the purines are of only two types. First is the adenine, second is the guanine. Now to avoid the confusion or to remember, in this spelling of pyrimids, the presence of Y. So the bases that are having Y will be our pyrimidine bases. So cytosine there is Y, in thymine also there is Y, so in pyrimidine is Y. So the bases are the thymine and cytosine, and we can remember the uracil. While in purines, only adenine and guanine are present. Now we will proceed with the structures of the purines and pyrimidines. So we can see these structures are already drawn here. Now we will see one by one. So this first structure, this is the uracil. We can see that at here we will numbering it. We will start the numbering. This is our first carbon, first nitrogen, second, third, fourth, fifth, and sixth. These are our pyrimidines. Here this is our pyrimidines. And these are our purines. Pyrimidines are the single ring and purines are the double ring. So in this pyrimidines, the nitrogen is present at the first and the third position. So this is the uracil, and uracil is only present in the RNA. We have to remember that uracil is only present in the RNA. So in this uracil, when there is addition of methyl group at the fifth position, we will get the thymine. So we can see here the structure is basically same, only at this first, second, third, fourth, fifth, and sixth. At the fifth position, there is addition of this methyl. So this is the thymine. There is another name for the thymine. Thymine is also called as the 5-methyl uracil. Thymine is also called as the 5-methyl uracil because in the uracil at the fifth position there is addition of the methyl group. After thymine, this will be our cytosine. This will be our first, second, third, fourth, fifth, and sixth. Remember, in the cytosine, at the sixth position, there is presence of NH2 group. While in this both structure, there is no NH2 here. Double bond O is remaining. Yes, now the complete structures. So this is the pyrimidines. After pyrimidine, now let's move to purines. So this is the purine. The purine is made up of 9 members. Now let's start the numbering. This is the first, second, third, fourth, fifth, and sixth. Here it is the seventh, here 8th, and here 9th. So this is the 9-member structure. At the position 1, 3, 7, and 9, there are the nitrogen present. In the purines, at the position 1, 3, 7, and 9, the nitrogen are present. And here also we can see 1, 2, 3, 4, 5, 6, 7, 8, and 9. In this also, at the 1, 3, 7, and 9, the nitrogens are present. Now let's see why I have written this nitrogen in the red color. So wherever there is this red color nitrogen, at that position the bond is going to form. This nitrogen is going to form the bond with the sugar molecule. So at this first molecule of the pyrimidine, the bond is going to get formed, and at the 9th position of the purines, the bond is going to get formed. So I hope all the structures of this pyrimids and pyramids are clear. Now after this, we are going to see how a nucleotide is formed. The basic structure in the nucleotide is composed of sugar plus nitrogen base plus phosphate group. But when there is only presence of sugar and nitrogen base, it is called as the nucleoside. And this nucleoside plus phosphate will give nucleotide. This is very important to remember, that the sugar plus nitrogen is called as the nucleoside. When there is addition of phosphate group in the nucleoside, it will give us the nucleotide. Always remember. Now we will see how these bondings are formed. The bonds are formed between the various sugar molecule, nitrogen molecule, and the phosphate molecule. So first of all, there is the sugar. We know that in the DNA, the sugar present is the pentose sugar. So we will draw this sugar, pentose sugar. The fifth carbon is present outside the ring. This will be here CH2, and here will be presence of OH group. So this is our pentose sugar. Now after this pentose sugar, we will have to draw a nucleotide. A nitrogen base. So we will, for our easy explanation, we will take the thymine. So this is our first this is our first nitrogen, second nitrogen, third nitrogen at the third place. So this is our thymine. This is our pentose sugar. Now here is hydrogen atom is present. There is no need to mention this hydrogen atom, but for now, for our understanding, we have to consider this hydrogen bond. So now, when the bond is formed between the sugar and the nitrogen, let's name it. This is the sugar and this is the nitrogen base. Whenever there is the formation of bond between the sugar molecule and the nitrogen molecule, there is the release of the oxygen molecule. There is release of the water molecule. So this OH molecule from the sugar and this hydrogen molecule from the nitrogen base, they are going to get released as the water. There will be water will be released out as this OH plus H. This will form H2O and the water will get released. After their release, the bond that is formed between the nitrogen base and sugar molecule, this is the bond that is formed between this both. Now this bond is called as the N-glycosidic bond. The bond formed between the first carbon of the sugar molecule and the third position and the first nitrogen is called the glycosidic. The bond formed between the first carbon and the first nitrogen of the nitrogen base is called the N-glycosidic bond. Here N stands for nitrogen and glycosidic, here glyco means sugar, which is the pentose sugar. So N-glycosidic bond. The bond formed between the sugar molecule and the nitrogen base is called as the N-glycosidic bond. Now this is our first bond. So our nucleotide has been formed. The sugar and the nitrogen base, that together form the nucleoside. So our nucleotide has been formed. Now in this nucleotide, when there is addition of the phosphate group, our nucleotide will be formed. Now let's see how the bond between the sugar molecule and the phosphate group is formed. So this is our phosphate, double bond O, OH, OH and OH. This is the structure of phosphate group. So always remember that from the sugar molecule, we are going to get OH molecule. So this OH molecule from the sugar and this hydrogen molecule from the phosphate group, they are going to form the bond and they will be released as the water. And so after the release of the water, there is the formation of the bond. Now this water will get removed. now this is the formation of the bond between the phosphate group and the sugar molecule. Now this bond is called as the phosphoester bond. The bond formed between the fifth carbon of the sugar molecule and the oxygen molecule of the phosphate group is called as the phosphoester bond. Always remember that phosphoester bond is formed within the nucleotide. The bond that joins the oxygen molecule of the phosphate and the fifth carbon of the sugar molecule is called the phosphoester bond. Now this is our one nucleotide. Now this similar nucleotide will be there will be another molecule of sugar, phosphate, and here maybe another base will be present such as adenine, guanine. So they will be present. Now let's see. This is for our phosphoester bond. The bond between the one sugar molecule and the other between the one nucleotide and other nucleotide is called the phosphodiester bond. So phosphodiester bonds it's connected to nucleotides, while the phosphoester bond is between the nucleotides. So here, let's suppose I am only drawing the sugar. This will be here nitrogen base. That's too far away. This is our sugar, and this is our nitrogen base. Now there will be formation of the phospho. So this is the phosphoester bond. Remember, the phosphoester bond is between a nucleotide. Now, this is our second nucleotide. It is having sugar, nitrogen, and the phosphate group. Now, from this OH is going to get removed from the third carbon. So this is our first, second, third, fourth, and this is our fifth carbon. So now this OH from this and water molecule, they are going to get released as the H2O. After the release of H2O, there is a formation of bond. This bond is called as the phosphodiester bond. Forms the bond between the oxygen and the third carbon of the. This is known as the phosphodiester. This is the 5' end, as this fifth carbon is here, so this will be our 5th prime end, and this will be our 3' end. So let's see. So let's remember, this will be our 5' end, this will be our 3' end. Now let's have a quick revision on the type of bond that are formed. The first bond is the N-glycosidic bond. This bond is formed between the sugar and the nitrogen base. The carbon that is the first carbon, and the nitrogen is also at the first nitrogen. The bond between the first carbon and the first nitrogen. The second type of bond is the phosphoester bond. Now phosphoester bond, it is formed between the sugar plus phosphate group. This bond is formed between the 5th carbon and the oxygen of the phosphate. And the third type of bond that is formed is the phosphodiester bond. Now this phosphoester bond, it is formed between two nucleotides. and the carbon that is involved is the third carbon, and between the oxygen. So there is no mention. So these are the three types of bond: young glycosidic bond, phosphoester bond, and the phosphodiester bond. I hope all the bonds that are present in the DNA are clear to all of you. After this, we will see now the double helix model of the DNA. Double helix structure of DNA. This double helix structure was given by Watson and Crick in year 1953. Now we will see the features of the double helix model one by one. So, double helix model was given by the Watson and Crick. Now, the first feature of this double helix model is that DNA is composed of the two nucleotides of the two strands. So now let's draw the strands. This is our first sugar. This is our first strand. Now we will see another strand. This is our two strands. So the polarity of this strand will be 5 prime. And so if this is 5 prime end, this will be our 3 prime end. So obviously the second feature is that both these strands of the DNA run anti-parallel to each other. Meaning that if this strand is from 5 prime to 3 prime, then this strand will be from 3 prime to 5 prime. Now, the bases that are present in the DNA are projected inwards. So here there will be our bases. Let's add one more sugar here. The bases will be present will be towards inside, and the phosphate will be present outside will be projected outward. Phosphate. So this will be the sugar and the phosphate forms the backbone of the the DNA molecule. Backbone of DNA is formed by the sugar and phosphate. Always remember, the backbone of the DNA is formed by the sugar and the phosphate molecule. This will form the backbone of the DNA molecule. So now these bases are always paired inward. So now let's see how these bases are paired. Always remember that pyrimidine will form a bond with purine. Always one purine will form a bond with other pyrimidine. There will be no like this purine in one strand and other strand also there will be purine. No, the bond that is formed in the DNA are only pyrimidine and purine. The bonds are formed between the purines and the pyrimidines. So let's see. The next feature is that the bonds that are formed between the nitrogen bases are adenine will always form a bond with thymine, and cytosine will always form a bond with guanine. So adenine and thymine, cytosine and guanine. This is thymine, this is the pyrimidine, this will be the purine. Cytosine, this is our pyrimidine, this will be our purine. So the bonds are formed between the purines and the pyrimidines. So now let's see here A, T, G and C. So in the opposite strand to the adenine, there will be thymine, as adenine will always form a bond with thymine, AT. So thymine will form a bond with adenine, guanine will form a bond with cytosine, and cytosine will form a bond with guanine. Now these nitrogen bases are attached to each other with the help of the hydrogen bonds. So the hydrogen bond between the adenine and thymine is a double bond. Adenine pairs with thymine with the help of double bond. So A double bond T. So obviously thymine will also form a bond with adenine double bond. Now the guanine and the cytosine will have a hydrogen bond that is of triple bond nature. So triple bond. The bond between the guanine and cytosine will be of 3 types, and similarly here also. So these are some of the features of the DNA double helix DNA model. So now let's see the diameter and the distance between the nucleotide bases that are present in DNA. So the diameter between this is of 20 angstrom units. So the diameter of the DNA is 20 angstrom units, and the distance between two nucleotides is of 3.4 angstrom unit. The pitch of DNA is 34 angstrom unit. What is the pitch? Pitch consists of the 10 nucleotides. Roughly 10 nucleotides are present in a one pitch. So these are the features of the double helix model. So now let's draw a double helix here. Now let's see, there are two strands of the DNA. So one strand, the polarity of this strand will be 5', and this goes here, and this will be a 3' end. So if this is 5' end, this will be obviously a 3' end, and this both strands are anti-parallel to each other. So this is 3', and here it will be our 5'. There will be presence of the bonds. Here will be adenine, whatever, the sequence can be any. Adenine A, T, G, C, A, C, C, G, A, T, G, A, C, 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 T, A, C. So this will be the bonds between the hydrogen and in the nucleotides will be present. This is called as the pitch. It contains roughly of 10 nucleotides, and this pitch is of 34 angstrom unit. Now it will be clear that the 10 nucleotides is considered of one pitch. So this will be of 34 angstrom unit, and between these two, the distance between these two nucleotides will be 3.4 angstrom unit. Between this, and the diameter will be 20 angstrom units. So this completes our helix model that was given by the Watson and Crick in the year 1953. I hope every one of you understood the lesson. Thank you.